Thank you for the invitation. I'm bummed that I needed to be out of defense today and then teaching, but I'm really looking forward to seeing some of the recorded talks. What I'd like to talk about is SARS-CoV-2 and modeling the effects of prior immunity and using kind of a viral population perspective. And you'll kind of see where that comes from. But I want to start off with a little bit of a disclaimer. I think much more about kind of the virus, its evolutionary dynamics, its population dynamics, and so forth, and tend a little bit to kind of black box the immune response. I was excited to be invited to this workshop mainly to learn from you guys about where some of what I'm thinking is kind of going wrong, basically. Let me move this little screen. The main thing I'd like to talk about today, and they're very, very simple models that I'd like to discuss, is how do we understand the effects of prior immunity through either natural infection or through vaccination. And I'm thinking primarily of kind of antibody responses, humoral responses. So how do we understand the effects of that prior immunity on future kind of susceptibility to infection, right? And its effects on within host viral dynamics. And of course, I'm thinking about Jane's slides in the very beginning of this workshop, where was being shown was kind of that prior immunity, or immunity kind of affects infection potential, but also affects, onward transmission potential, right? So how does a prior immunity affect susceptibility to infection within host viral dynamics, that impact then onward transmission potential, and then ultimately also symptom development? And I'm not really going to talk about symptom development much, but really focusing on susceptibility to infection within host viral dynamics. And again, I'm thinking about kind of prior immunity in the context of SARS-CoV-2 through either natural infection or through vaccination. Okay, so and I said I was going to take kind of a viral perspective here. So I'd like to talk a little bit about some work of ours we've just recently published, which was kind of for the SARS-CoV-2 timeline of things. It took forever in terms of editorial process, but effectively this work that we've done is looking at looking at the viral population and asking a question of how much virus is actually being transmitted from a donor individual, so from someone who's infected SARS-CoV-2 to an individual who's kind of considered the contact or the recipient of the infection. We're asking questions about kind of the transmission bottleneck. So we can imagine that over here we have the donor with all these little marbles in here of different colors. So those could be different genetic variants, for example. So we have this parent population, and then during kind of transmission, right, we have kind of this bottlenecking, right? So we have these surviving individuals that really establish genetic lineages kind of in the recipient host, right? And so we're interested in understanding or quantifying the transmission bottleneck size, which we'll call NB, right? Which is just the number of virions that go on to establish a genetic lineage in an infected individual. But here, it's not 2 here. Here it would be on the order of, 10 or something like that, would be the transmission bottleneck size. So it's not the number of virions that fall on, the recipient, but it's also not the unique number of virus genotypes that establish infection, but it's this kind of population size that, of viral particles that goes on to establish genetic lineages kind of in the recipient host. Okay. How are they estimated? So we've been interested in this for a few years now, before we even knew about SARS-CoV-2. So the idea here is that we have a donor individual, right, with a sample taken from that donor individual, right, when their viral load is pretty high. We can deep sequence that viral population, and then we can kind of go across the genome and call variants at certain frequencies. As you can see here, maybe this green variant at this site, is above the threshold of detection. Maybe it's at 5 or 6% frequency or 8% frequency, and here this blue variant maybe at 45% frequency, right? This donor individual is going to transmit it to a recipient. That viral population that founding population, right, is going to kind of grow in the recipient, and then that recipient will be sampled at some point, right? And the same thing can be done with respect to deep sequencing the sample. and calling variants, right? So the idea here is if we have an epidemiologically linked transmission pair, so we know that there's a donor and a recipient, and we know who's the donor and we know who the recipient is, right? Based on these variant frequencies, what we are interested in is estimating the founding population size, NB. And so, excuse me. So we've established this method called beta binomial method. It incorporates various things like a stochastic birth-death process here in the recipient. It statistically accommodates the variant threshold, variant calling threshold here, et cetera. And if you're interested in this, I'd point you towards this, this paper in Journal of Virology. So what's the transmission bottleneck size for SARS-CoV-2? So previous work that had been done in seasonal influenza by Adam Loring's group in particular, has found that the bottleneck size for seasonal flu is tiny. It's 1 or 2 viral particles, so very, very small. So we were interested in seeing what the transmission bottleneck size was for SARS-CoV-2, and this was some of the initial work that kind of came out. This came out of a group in Austria, and what they had was incredible data where they had 39 epidemiologically linked transmission pairs. And you could see those transmission pairs here. The first number is the identifier of the donor, and the second number here is the identifier of the recipient, right? So we've got 39 of these. Here's the bottleneck size estimate they came up with using our beta binomial approach. And they had a variant calling threshold of 1%, so very, very low. The conclusions they came up with was that there was a lot of variation, right? But that many of these transmission pairs had on the order of about 1,000 viral particles as the transmission bottleneck size, right? Which is pretty large, especially compared to this 1 or 2 viral particle estimate for seasonal flu. And it led to a lot of discussion in the literature about, could transmission bottleneck size be an indicator of symptom development, right? So if you get infected because you're talking to someone for an hour really closely versus just a couple minutes where, talking to them closely for an hour might transmit more virions, right? And then might lead to more severe symptoms, so there were discussions like that were kind of had, and that was especially because these bottleneck size estimates were so large, and that there was so much variation between them, right? So what we decided to do is actually kind of look back at this data set, just because it was so different from seasonal flu. And we were surprised by that because it's an acute infection like seasonal flu, and even though it's not, as kind of well adapted yet, to humans, presumably, this is kind of early on, its R0 was, actually higher than for seasonal flu. But we just want to kind of look at it given how similarly these viruses actually transmit. I want to, it's kind of late in the day and a lot of people are kind of, I think, probably phasing out at this point in time. So I wanted to get a little bit of user input here. So a thought experiment, okay? So an example to provide intuition. So let's say we have a variant that's at a certain frequency in the donor, let's say by, this area here. So it might be at 45% frequency in the donor. Right? So it could be like this blue variant over here at the site. If there's a very large transmission bottleneck, what is the frequency that we would expect in the recipient? Don't all answer at once. It's supposed to be a workshop, right? Come on.
Should be the same, right?
Yeah, exactly. Who said that?
Duncan.
Oh, great. Thank you. Okay, yeah. So there's a large transmission bottleneck. We expect the frequency of the donor to be mirrored in the recipient, right? Because basically, if you're sampling a lot of particles, right, the recipient is sampling a lot of particles from the donor, right? The genetic composition of their population, their viral population, should look similar to that of the donors, right? So if the bottleneck size is large, we expect kind of the variance to fall along this one-to-one line, line, right? Over here. What about if the bottleneck's small? let's say it's on the order of 1 viral particle. Okay, I'll tell you. So if it's on the order of about 1 viral particle, right? If it's at, let's say, 45% frequency here, and it's just a matter of kind of chance, right? And kind of genetic drift, there's going to be kind of, a 45% chance, right? Or a 55% chance that you're not going to see, that you're going to, that it's going to be kind of lost in the recipient, right? Sorry, there's going to be, if it's at 45% in the donor, right? There's a 45% chance that it will not be transmitted, right? And a 55%, sorry, it's been a really, really long day. If it's a 45% chance, 45% frequency in the donor, right? Then there's going to be a 45% chance that it's going to be transmitted to the recipient, and in which case it's going to be up here at 1, right? Because all of the viruses will have that in the recipient, right? But there's going to be 55% probability that it's going to get lost, right? And so you're going to see it at 0, right, or below the limit of detection, right? And so what you expected with a small bottleneck is all these variants that you see here, either at 0 or at 1 in the recipient. With, if it's at higher frequency in the donor, it's more likely going to be at 1, right? And so what we decided to do was look at these kind of plots of donor variant frequencies against recipient variant frequencies in these transmission pairs that in the Papa et al. Study. And this was done with a graduate student in my group, Mike Martin. And this is kind of a comment that we just kind of published on based on the previous paper. And what you can see here are 2 things. One is, very low frequencies. This is up to 6%. We have, these variants that are on this 1-to-1 line, right, which are telling the story of kind of a very large transmission bottleneck, right? But then we have these kind of higher frequency variants here in the donor that don't look like they're being transmitted at all, right? So these 2 variants, there's different variants that are telling very different stories, right? And so we did a bunch of kind of analyses of these, variants and so forth. And one thing that we found, which was kind of the most direct evidence, is something that these very low frequency variants were actually spurious, and that we should pay attention to kind of these high frequency variants. First of all, high frequency variants are much more likely to be kind of true, right? But then what Mike did was he looked at those low frequency variants that appeared to be shared, at very similar frequencies. And he actually found that the overwhelming majority of samples, regardless of whether they came from Western Austria or Eastern Austria, but that they were found in a lot of the samples, right, at very, very similar frequencies. So you can see this one, this variant was found in all 43 samples that we looked at, and O is between 1% and 2%, right? And so you can see this kind of across all these kind of variants here. And this is just in the order of kind of how frequent the variants are. So this first one's in 43 samples, this one's in 43 samples, et cetera, right? This one's in 37 samples. So we think that these are spurious, related to kind of something related to kind of the sequencing protocol. And so what we did was we increased the cutoff above kind of, we looked at all of these variants that were found in multiple samples, and we increased the variant calling threshold to kind of above the highest value here. So we increased it to 6%, which is kind of a very, coarse sort of way to do it. But what should happen is it should give us kind of, very large confidence intervals, but it should at least tell us kind of what our maximum likelihood estimate is for the transmission bottleneck size once we know we've excluded these what look like spurious variants, okay? And so what you see here is the data organized by the maximum donor allele frequency. So basically in that plot, this is the variant frequency on the x-axis that is the variant that has the highest frequency in the donor, right? And what we find here is going from 1% cutoff to 3% cutoff, and then finally to the 6% cutoff, that we have kind of this iterative kind of decrease in terms of the transmission bottleneck size that's estimated, right? And that's because as we go from 1% to 3% to 6%, we're effectively cutting out some of those spurious variants that are very, very low frequency, right? That are basically putting weight on there being a very large transmission bottleneck size, right? And so what you can see is with a 6% cutoff, all of these, with the exception of this one, all these transmission pairs indicate that there's very likely kind of just a one to two viral particles getting across, really, and being responsible for establishing infection in the recipient. So combining this kind of across these transmission pairs, just to get a more robust estimate, what we find is by fitting this kind of zero truncated Poisson distribution, that on the order of about 80% or so transmission events, we expect it to be seeded by only about one virion. In about 15%, it's two, and so forth. So that over 99% of transmission events are seeded between 1 and 3 variants, viral particles. So we think the bottleneck size is also, is very consistent with that of influenza, seasonal influenza, and very, very small. Okay, so how does this get to immunity? Sorry for the long digression. So how does this get to immunity? So many of you are probably familiar with this expression here, which is from kind of population ecology. If we think about kind of a spillover event, the probability of a successful invasion occurring is just 1 minus 1 over R naught to the N, where N is the number of kind of spillover events, okay? So we can think about this in the context also of a infection, right? Where N is really the number of viral particles, right? And R naught is the within host R naught, right? And so what I have here is just the within host R naught shown here on the x-axis. This probability of successful infection given by this equation over here. And with a very small transmission bottleneck, here, for example, with kind of an n equals 1, and here with an n equals 3. So this is with n equals 1, this is with n equals 3, right? And then some estimates of kind of R naught from a ferret study of Katia and Parelson's. So here what you see is, of course, kind of with a higher within host R naught, the probability of successful infection is higher, right? And of course, kind of with a larger number of viral particles, that are being kind of effectively transmitted, the probability of successful infection is also higher. Okay. So now if we think about kind of immunity, right? So let's say we have a classic kind of target cell limited model that all of you, I'm sure, are familiar with, right? So we have this model here from Katia et al., where we have uninfected cells, infected cells, infectious virus, non-infectious virus, right? And we can just write out kind of what the R naught is for this target cell limited model, right? If we think about prior immunity now, right? If we think about prior immunity, we can think about kind of that being initiated by antibodies, right? And so we can add kind of on these terms on this equation over here and also on this equation over here, which is just another clearance term of the free virus, right, by antibodies, right? And so it's this minus kappa AVI term over here and minus kappa AVN term on this equation, right? And the R naught is just changed over here to look like this, right? So it adds this term over here. It increases the denominator, right? And so by increasing the denominator, what it does is it decreases R naught, right? So if we think about, let's say, this n equals 1 line, right, for one viral particle, if we, let's say the within host R naught is on the order of about 2, right? If the antibodies that are circulating, are increasing clearance, right? What we expect is that the probability of successful infection, is going down, right? Which makes sense. I mean, this is consistent with what we know about vaccination. If we're vaccinated against SARS-CoV-2, we're less likely to get infected, right? So prior immunity, antibody-mediated immunity, protects against infection. So it reduces susceptibility to infection. And we can think about this as happening by lowering within host R naught, right? So that's pretty basic, right? And so if we think about predictions from kind of this viral dynamic model where we have these additional antibody terms, right? we can think about kind of the target cell limit model, for example, here being in black, where we have infectious virus, all virus, and antibodies, right? And going from the black to the one in red, we think about this as kind of a primary infection. So let's add on these expansion of antibodies, right? And so we go from these black curves to these kind of red curves over here, right? If we're incorporating an antibody response, right? Which results in fewer target cells actually being depleted, et cetera, right? And faster clearance. Okay, so now we have this primary infection model with antibody response, right? And now what we can think about is kind of a secondary infection, right? Once we actually have that antibody response, right? And so we've already known from kind of these slides, over here, that if we have some antibodies that are kind of circulating, it decreases within host R naught, right? And it reduces the probability of successful infection, right? But then in breakthrough infections, right? Let's say prior immunity is from natural infection or from vaccination, right? Secondary infections, which are breakthrough infections, right? That have this kind of antibody response, you start off with kind of a higher level of antibodies, right? A decrease in the within host R naught. So what we would expect just by kind of intuition, right, is with a lower within host R naught, we expect, we can see it here with infectious virus and all virus, we expect a slower rise in viral load, right, because there's a lower R naught, right? So a slower rise in viral load than in primary infection. We also expect a lower viral peak, right, that you can see here and here in comparison to this red one. And then we also expect a little bit of slower viral decline, right? So those are expectations. But then the thing that kind of baffles me is this really wonderful work that Stephen Kistler and Jonathan Grad have been working on, where they have data from NBA players, right? That are being where there's just incredible surveillance, right? And they're getting tested very, very often to see if they're positive, right? And so some of these players were vaccinated, some were unvaccinated. And here's some dynamics of just those individuals. So this is, plotting all these individuals kind of on the same plot. Here we have CT, which a lower CT is a higher viral load, right? And here, this is days since peak viral concentration, right? Just to kind of standardize these two. And what you see is in unvaccinated as well as in vaccinated individuals, right? You see kind of this this kind of rapid rise, right? And then of course this decline, right? And they've looked at this quantitatively by fitting kind of lines to these data, consistent with exponential growth and exponential decline. And what they found is that the time from kind of infection to the time of peak, so the proliferation time is similar between unvaccinated individuals who got infected and vaccinated individuals who are experiencing breakthrough infections. The peak viral concentrations are very similar also, right? But the clearance time is faster for unvaccinated individuals compared to vaccinated individuals, right? So, in the spirit of this being a workshop, I wanted to just kind of end with some questions and discussion, so at least the way I'm thinking about prior immunity, reducing susceptibility to infection, I think about it as, reducing within host R naught, right? Because some of that virus is being cleared by existing antibodies, right? And within host R naught, if there's very few virions found in infection, that decreases the probability of a successful infection taking off, right? But if antibodies do effectively decrease within host R naught, then shouldn't we also expect viral load to increase more slowly? So have a kind of a slower proliferation rate, right? And then shouldn't we expect it also to peak at lower levels rather than similar levels? So neither of those things are observed, right? And we might also expect viral decline to be slower in vaccinated individuals than in unvaccinated individuals, based on those, very simple simulations. But this is again what's observed, against what is observed. So I just want to kind of end with a question of, how do we understand the observed within host dynamics in individuals with no versus some prior immunity, simultaneously with kind of this reduction in susceptibility of vaccinated individuals that presumably acts through kind of decreases in within host R naught. And with that, I think I'm right at my end also. So I'll leave you with that. Maybe you guys can help me answer some of those questions. That's it.
Thank you. The questions disappeared, but does anyone have answers?
Well, I was wondering if you could think about it in terms of, because it's individuals who are having breakthrough infections that these observations are in, right?
Mhm.
And so these are individuals that probably don't really have that neutralizing immunity anymore, those antibodies. And so that won't really, so that means that their increase in viral load will be at the same rate. But then once the T cell immunity kicks in, then that's why they have a shorter infection.
Yeah, that's nice. I mean, that would require, I think that's a really nice explanation. I think that would require, I guess, a model of that individuals who experience breakthrough infections are somewhat, in some ways, different from ones that don't, right?
Mm-hmm.
Which very well could be the case. I mean, I don't know about, measurements of, antibody titers or anything like that, from the NBA players, right, to actually see whether those were predictive of where the breakthrough infections actually happened. But if we think about kind of that, just kind of a stochastic event of kind of who gets infected and who doesn't, based on that probability of successful infection, right? That wouldn't explain. So I agree with you that individuals who experience breakthrough infections would have to be somewhat different, right, in terms of their antibody levels or their immune response, compared to the individuals who do not. I don't know if there's any evidence for that though, and I would love to see that. Yeah, that's a really great point.
Okay, thanks. I think we might have another minute for another answer or question, I guess. I don't see anything in the chat, so.
I think Dick has a question.
Oh,
yeah, I don't have any answers, but I do have a quick question. I just wonder how the lower level of diversity within host for SARS-CoV-2 affects your estimates compared to, say, like the common cold or the other viruses you're looking at.
The lower levels of diversity, sorry, say it again.
So within a host, the population diversity is lower, if I understand it correctly, in SARS-CoV-2 than you would probably expect in the flu or other viruses you've looked at. So does that affect
there's actually very little diversity. Actually, this is some really, really nice work of Adam Loring's. He has a paper with it's a Macron et al. Paper in eLife. And they show kind of in seasonal flu cases that there's actually extremely low levels of genetic diversity in flu infections, seasonal flu infections. And you might be familiar with some previous work prior to that Loring paper. It was a Poon et al. Paper in Nature Genetics, which showed that there was a huge amount of genetic diversity in flu. And we actually when we developed our beta binomial approach, we actually used their data and came up with large bottleneck sizes based on that, as an application too. But it turns out that that there's a really nice once the Loring paper came out, it was this discrepancy, like, is the bottleneck size actually small or is it large, and how much genetic diversity is there actually in these infections? And one was a Hong Kong study and one was a, in Michigan study, but like, why were these findings so different? And there was some really great detective work on part of Catherine Shah and Jesse Bloom. And they have a comment to the Poon et al. Paper. So basically what they found through looking actually at the read files, was that there was this, these paired end reads were actually like not being mapped appropriately. There seemed to be what they call technical contamination. So it seems like it was actually like computer code that really jumbled up kind of the reads, and therefore kind of put sequences, put reads kind of into kind of samples that they shouldn't have. And so basically basically a lot of these samples share diversity because of that technical contamination, you know. So I'll point you towards that paper. So I think the Poon et al. Results are not really valid. And actually, Poon et al. Went back and resampled some of their samples and using a different method and so forth, you know, and they also agreed that there's very low diversity.
Okay, well, thank you. I'm out of the literature, I guess, too far.
Yeah, you can follow that string of kind of back and forth in the literature.
I look forward to doing it now.
I don't know if there's time. Maybe we can, I know I'm a little bit over, so maybe Chapin, maybe we can wait to panel. I don't know.
Sure, I can lower my hand, that's fine.
Okay. Yes, so our next, so thank you very much.